So hello friends, welcome to Medicine 101. Okay, so I'll talk about two of the pathways of apoptosis in terms of the extrinsic pathway and intrinsic pathway. So before starting the topic, let me tell you guys, I have uploaded on video where about the apoptosis. Okay, where I have just said something related to this part. So it's my humble request to you guys, please watch the video first, then just come to this video. If you don't watch, then maybe you will not be able to connect in between these two videos. Okay, so for having a better understanding, please do watch the first video of apoptosis that I already uploaded in my YouTube channel. Okay, then come to this one. Okay, the pathways. So if we just start this one in terms of the extrinsic and intrinsic pathway, we will get the two types of the pathway I have already written over here, the extrinsic and intrinsic. So first I will talk about the intrinsic pathway over here. Okay, so let me write it over here, that intrinsic pathway. Okay, so within the intrinsic part, whatever the things we will see over here. See, in my first video I have already uploaded that, for example, if I just consider this is the mitochondria. Okay, just like this, and let me just draw just like this. Now, within the mitochondria, there are presence of what? Cytochrome C for oxidative phosphorylation. There is the presence of what? C. Under normal circumstances, what do we know that there is presence of certain types of the genes or proteins or channels, whatever you can say over here, that is known as what? BCL2. Now, for example, is the BCL2. Under normal circumstances, this BCL2 will not let the cytochrome C to go outside of the mitochondria. They will be produced over here. Okay, and this BCL2 gene that depends on certain types of the this BCL2 gene depends on certain types of the growth factors. Certain types of growth factors. Now see, if the growth factor is normal, then the gene will work normally. But whenever we will see that for any region, if we see the deficiency of the growth factor, we will see this gene will not work properly. Instead of that, we will see certain types of the genes that will be active. Okay, for example, let me show over here, just like the BAC or BAX gene, BAC or BAX gene, and whenever these genes will be activated over here, they will allow the cytochrome C to go outside of the mitochondria. So the cytochrome C, they will come outside. They will come outside of the mitochondria. Okay. Now what type of the thing we'll see after that? After coming the cytochrome C outside of the mitochondria, they can activate certain types of the factors that is known as APF. That is what? Apoptosis protease activation factor. So if you write it over here, apoptosis protease activation factor. Activation factor. Now what we will see after that? At first we will see this can activate certain types of the what? Caspase. It can activate certain types of the caspase. Okay, caspase. What is caspase? If we see the caspase over here, it's the cysteine containing one type of the protease enzyme. Cysteine containing, cysteine means one type of the amino acid we know over here. Okay, sulfur containing. Now, what is it? Caspase is it's what? A cysteine containing. Cysteine contain. We can write over here. Cysteine contain. Okay, protease enzyme. Protease enzyme over here. Now what is over here? If we just write it this term over here, caspase. Okay, whenever let me write it over here, caspase. Now see. Okay. Now whenever they will see that the presence of the aspartic acid in anything, they will just found this molecule. So cutting in terms of what? Aspartic acid. Okay, how we got it over here. Cases we will see there will be activations of the certain types of the caspase, but what is the first caspase that will be activated, or we can say the initiator caspase. The initiator caspase we will get it over here. There is the caspase 9. So initiator caspase, that is what? Caspase 9. Caspase 9 over here. Okay, at first we will get. Now this caspase 9 that can activate certain types of more caspase, that is known as executioner caspase. Okay, let me write it over here. That is known as what? Executioner caspase. Okay, executioner caspase over here, and here we'll get caspase 3 and 7. This type of the caspase. Now this type of the caspase can activate two types of the enzymes. One is what? Protease, another is endonuclease. So whenever we will see this executioner caspase, they will just what? They will activate the protease. Along with that, they can activate the endonuclease. So whenever they will just activate the protease enzyme, they will destroy the structural proteins of the cell. Structural proteins. And what about the endonuclease? They cut the DNA. Cutting of DNA. So as a result, whenever we see what? The structural proteins of the cells is getting destroyed, along with the DNA is not, there is no existence of the DNA. We can say what? The cutting of the DNA, ultimately there is no chance for the cells to survive. So ultimately we will see what? The cell death. We will see cell death over here. Okay, cell death. Now we'll connect the extrinsic pathway with the intrinsic pathway with the extrinsic pathway. Now whatever the thing we'll see in terms of the extrinsic pathway. Extrinsic means something that will come from outside. Okay, let's suppose there is a cell membrane. There is a cell membrane. On the cell membrane there is presence of certain types of the receptors over here. Like this. Now cell membrane. This, okay. Now it's a cell membrane over here, okay, and these are the receptors. Now these are the known as what? Phase receptor, first. So let me write it over here. Phase first receptor, or you can say TNF receptor. TNF receptors. Now see, whenever any cell, whenever we know that whenever any cell will get injured, at that time they will what? Secrete different types of the chemicals. One of them is what? Phase L. That means phase ligand. So during the cell injury, let me write it over here. Cell injury, you will secrete the TNF alpha, or they can secrete the phase ligand over here, and we will get over here on the in the bottom of the receptors we will get fat. Okay, let me use this one over here. FADD. Now what is FADD? FADD stands for phase associated death domain. Phase associated death domain. Okay. Now see, whenever these things will be activated over here, there is no chance for the cell to survive. How these things happen? So I will just talk about these things in a couple of minutes. Okay. Now see, whenever the TNF alpha, the phase ligand, these things will be secreted. Whenever the cell is getting injured at the time, this phase ligand will come over here just like this, and they will, for example, they will just get binded with the 3 receptors over here. So as a result, we will see trimerizations. We'll see what? Trimerizations. The receptors over here. Trimerizations. That means 3 things will get binded together. Okay. The 3 receptors will get bound with the word phase ligands over here. And as a result, we will see whenever this phase ligand will get attached with the receptors, this PAD, that means phase associated death, will be activated. And whenever they will be activated, they can stimulate the process of caspase activations. Caspase activation. And whatever the caspase they will activate, for example, caspase 8, okay, in terms of the animals. So the caspase 8 Okay, we can say in terms of what? Animals. And in terms of in case of the human being, we will see there is another type of the caspase that is known as caspase 10. This is the caspase 10. Now see, whenever this caspase will be activated over here, now they are having the capacity to directly activate the execution of caspase over here. I already said it over here. Now this can activate directly. Okay, let me show over here. Okay, if I want to show you in this way, just like this, it's better to write it over here. Already I have showed over here, I have shown over here what the executional caspase. So this caspase 8 and caspase, the 10, they are having the capacity to activate what? The execution. That means, okay, let me write it over here. Executional caspase. That means what we will get over here? 3, 6 and 7. Now they can activate what? The protease. The rest of the thing is same. And they can also activate the endonuclease. Here we see whenever the protease will get activated, they will just destroy the structural proteins of the cell. Whenever the endonuclease will be activated, they will just destroy the DNA. As a result, there is no chance for the cells to get survive. That will be cell death. Ultimately, we will get over cell death. Okay, we will get the cell death over here. Now there are some certain points about the apoptosis. The some characteristic features. Let me write it over about them. Okay. Yes. Yes. So, what are the features we will see in these cases? So one thing I thought to say that this phase, okay, this receptors actually we can say that cluster differentiation for 95. CD 95 we can see in these cases. Okay. Now we'll see the features in terms of what? In terms of the apoptosis. What are the features we'll get over here? Okay. So features over here. I have already said before, there will be what? Cellular size that will get decreased. Whenever the apoptosis will be seen in case of the cells, we will see the cell size that will get reduced, the shrinkage of the cell. So first we will write it over here. Shrinkage. That means the cell size that will be reduced or decreased over here. Okay, we'll see the cellular size that will be decreased. And as we know, in these cases, there will be no problem. Sorry, there will be no problem as far as the cell membrane is concerned. So cell membrane, there will be no damage on the cell membrane. There will be no damage on the cell membrane. So intact. We can write it over cell membrane that will remain intact. Okay. That means because why? There is no activations of the phospholipase enzyme. So if what we saw in terms of the what? These activations of the phospholipase we saw in terms of necrosis. But in case of apoptosis, we see there is no such activations of the phospholipase enzyme. So phospholipase, this enzyme, okay, this will be not activated. So as a result, there is no chance for the cell membrane damage. There is no chance for the cell membrane damage. And we will see what? Chromatin clumping, okay, so, or you can say chromatin condensation. It's better way to write clumping. Now, for example, if I show the DNA over here, for example, it's 3' to, okay, let me write it over 5' to 3' over here, 3' to 5'. Now, after that, we will see in this case, this will get fragmented for the activation of the endonuclease, and we will see partitions of this part, just like this. Okay, 3 to 5, 5 to 3, just like this in all cases. Okay. Now, whatever the thing we'll get in terms of the gel electrophoresis, because I already said in terms of the necrosis in the separate video that we'll get the smear pattern. Okay, smear pattern in case of the necrosis. Whenever we will see the cells under the gel electrophoresis. Now, if you see the apoptotic cells were in terms of gel electrophoresis, whatever the thing we'll see over here. So let me show the gel place. So in these cases, under the gel electrophoresis, whatever the thing we'll see. Under normal circumstances, if you see the cell, in these cases, there will be a normal band, just like this. Normal. But whenever we see the apoptosis of the cells, we will see in these cases, there will be step ladder fashion. This is known as what? Step ladder fashion. Step ladder, step ladder orientation, you can say over here. Orientation. Okay. Now we'll just see some clinical features where the apoptosis can be increased and some clinical features where the apoptosis can be decreased. Okay. So where we'll get the more apoptosis, where we'll get the less apoptosis, we have to know in this way. Okay. For that thing, let me erase this part. So let me show it over. There are more processes, but I'm just saying short. So the clinical situation for overaction of apoptosis. Okay, overaction of apoptosis. Where we will get more apoptosis, where we will get less apoptosis. Now we'll see the apoptosis that will be more in terms of the neurodegenerative disorders. Okay, if we see neuro, that will be definitely it will be more neurodegenerative disorders, or we can say in terms of the viral infections. In terms of viral infections, we'll get what? The more chances for the apoptosis. Now where we get the apoptosis less, we'll get the apoptosis less where? In terms of the autoimmune disorders. Autoimmune disorder. Here we'll get the apoptosis, that will be less. And another thing, for example, hope you have, all of you have heard the term P53 gene. So if we see the P53 gene, okay. Now, for any region, if we see the damage on the DNA, if we see for any region a damage on the DNA, then this gene P53 gene will do the apoptosis of the cell. That means it will cause the apoptosis of the cell. Okay. For any region, if we see there are the mutations at the level of P53 gene, if we see the mutations over here, then definitely that will cause the cancer over here. That will cause the cancer. At first, if you see the damage on the DNA for any type of the damage on DNA, then this P53 gene will do the apoptosis of the cell. Now, if you see the mutations on the P53 gene, then definitely there will be formation of cancers. Okay, the mutations of P53 gene that leads to formation of cancers. Okay, so that's all for the apoptosis in terms of the extrinsic, intrinsic pathway and the slide features of the what apoptosis. The clinical situations where overproductions or the less productions of the apoptosis, that means where can we see the more apoptosis, where we can see the less apoptosis. Okay, अगर ये वीडियो आप लोग को अच्छी लगे तो लाइक कीजिए, सब्सक्राइब कीजिए और ज़्यादा से ज़्यादा आप लोग दोस्त के साथ शेयर कीजिए ताकि उन लोग को भी इसका सुविधा मिले. ओके, थैंक्स फॉर वॉचिंग.